Welcome back, everyone. So for this week, I'm going to be talking about the BCA assay. So recall from the last experiment, we purified our beta-gal off of the anion exchange column, as well as the size exclusion column. So now that we have our purified beta-gal sample, before we can really perform any kind of experiment to test its activity, we need to know how much beta-gal we actually have. So to do this, we're going to be performing an assay that'll let us determine the protein's concentration in the solution. So there's a couple different ways that we can figure this out. The cheapest and the simplest way would be to use a UV vis. However, these results tend to be very unreliable, very nonspecific. They're mostly used to give us more of a ballpark range. Another option is to use fluorescence. So while fluorescence would be more specific by dyeing our sample with some kind of fluorescent light, it's also incredibly expensive. So neither the UV vis alone or using fluorescence would be the best approach. So that leaves us with two other assays. So one is a colorimetric assay like the Bradford assay, and another is a Biuret assay like the BCA assay. So both these assays actually kind of work very similarly. They let us determine the protein concentration based off of changes in color, while the Bradford assay is more of a direct approach where we specifically dye the protein. The Biuret assay is more of an indirect approach, and you'll see a little bit further in what I mean by an indirect approach. But over here you can kind of see what the BCA molecule looks like. It's a fairly large molecule, and what's most important about it are these two nitrogen groups. So like I said, the BCA assay is used to test protein concentrations based off of changes in color. We can see differences in concentrations when we analyze the colors using the UV vis at a wavelength of 562. And we basically set up some kind of standard curve, usually with some known protein like BSA, bovine serum albumin, and then compare this to our experimental, which would be beta-gal. So down here you can see what I mean by the standard curve. We generate some linear curve, and once we also have our experimental beta-gal, we should be able to follow this line and determine if it's, let's say, if it gave an absorbance of 0.6, then it should be this point, and that should mean that our beta-gal is about 700 micrograms per ml. So how exactly does the BCA assay work? So remember how I said that it was an indirect assay? So instead of just dyeing the protein, this assay is based on the reduction of copper 2+ to copper 1+. How does this happen? So the copper 2 is actually able to get reduced by amino acids in the protein, such as cysteine or tyrosine. And once this reduction happens, it gets reduced to copper 1+. Once this copper 1+ forms, it is actually chelated by two BCA molecules. And this formation of this copper 1+ BCA complex is what forms that purple color. So the more of these complexes that form, the darker the purple we're going to see in the solution. So this is how we can kind of determine that the more purple a solution is, the more protein there should be. So this is one of these experiments where both heat and time can kind of play key factors in this reaction. If we let this reaction sit at room temperature, it will slowly turn purple. But we don't really have the time for that, so we can also heat up the reaction and cause it to quickly turn purple to generate a really quick result of what or how much beta-gal we're working with. So this is normally how we set up the BCA assay. We'll usually generate usually set up some kind of standard curve, maybe 8, 10, 12 different standards, usually going from 0, 1, 2, 3, all the way up to however many samples we have. And this should be of protein, like a protein concentration that we would know. We would also do the same thing with our beta-gal sample, and remember, we don't know what the concentration of this sample is. By heating up our tubes for maybe 10-15 minutes, we will get some kind of color change, and we're able to compare our sample to this to the standards that we have previously set up, and we're able to determine that our protein concentration may be 6 mg per ml based off of how similar the colors may be. All right, so it's a rather simple assay to use to figure out the BCA, the protein concentration. So that's pretty much all I have to say about the BCA. But before I finish off the recording, I want to do a little bit of housekeeping. Ignore the new course drop deadline. So just remember that the drop deadline is on April 1st. So that's coming up within a few days. If you're not feeling confident that you're going to do well in the lab, please maybe consider looking into this. We only have 2 labs left after the BCA. So we'll be the next assay we'll be doing is the activity assay, and then finally the kinetics assay. However, I am thinking that we're probably going to skip the kinetics assay, at least the lab report, because setting up the kinetics assay is very similar to the activity assay. So instead, rather than just do a lab report, it would only just be a quiz on the kinetics assay. So once we finish these last assays, that'll be it for the lab. The last things you guys will need to do is just make sure you have your final presentations ready by April 25th. So I just want to talk a little bit about these final presentations. So these are going to be 15-minute long mock presentations that you're going to submit to me. What I want to see in these presentations is you being able to explain what happened throughout the entire lab from start to end. So what I mean is I want to hear everything you learned from the PCR lab all the way to the kinetics lab, and present it in a way that you would kind of present a poster of your research. So I want you to be able to kind of give the basic idea of what was done in each of these experiments and really highlight the theory. Show me why you use this kind of assay, why you performed this experiment, why did it work, why did you add things. Like, I need to know that. I need to know the why. I don't care too much about like methods and materials. You can pretty much skip that for the most part. But key components like why did we use DNA-SAB magnesium chloride during the lysis, or what was the T4 DNA ligase and why is it important? So try to think back of all the important pieces of the lab and try to talk about them in a relatively informative way. And what you'll just need to do is make sure to record your voice over the presentation. You don't need to do any kind of actual video recording. Just record the presentation and you talking over it, explaining what happened. And that's pretty much everything you need to do for the final presentation. But if you have any questions about about the presentation, about any of the labs, or the future of the lab, please feel free to email me and I'll try to get back to you guys as quickly as possible.